Between meals, during overnight fasting or during exercises, long-chain fatty acids are mobilized from triacylglycerols in adipose tissue. Triacylglycerols are hydrolyzed by hormone-sensitive lipase to fatty acids and glycerol. Once fatty acids enter into the circulation, they immediately bind to albumins, and in this binded to albumin form, they circulate through the bloodstream until they are uptaken by the cells. When fatty acid enters into the cell, it has to be activated by conversion to fatty acyl-CoA. Then fatty acyl-CoA must be transported from the cytosol into mitochondrial matrix. And the problem is that fatty acyl-CoA by itself cannot cross in a mitochondrial membrane, because mitochondrial membranes are impermeable to fatty acids due to their negative charge. To solve this problem, cells have carnitine shuttle. From fatty acyl-CoA molecule, carnitine acyltransferase 1 transfer fatty acid into carnitine molecule with formation of fatty acyl-carnitine, and carnitine has its own transporter on mitochondrial membrane. So fatty acyl-carnitine through carnitine transporter is transported into mitochondrial matrix. In the mitochondrial matrix, carnitine acyltransferase 2 enzyme transfers fatty acid from carnitine back to CoA molecule with formation of fatty acyl-CoA, which can be used in beta-oxidation for energy production, and the activity of carnitine shuttle is regulated by Malonyl-CoA that inhibits carnitine acyltransferase 1. The concept here is that Malonyl-CoA is one of the intermediate metabolites in fatty acid synthesis, and obviously as any anabolical process, fatty acid synthesis consumes energy. Malonyl-CoA is produced from Acetyl-CoA by an enzyme Acetyl-CoA carboxylase. Recall that this conversion is the rate-limiting step in fatty acid synthesis, and the activity of Acetyl-CoA carboxylase is inhibited by AMP-activated protein kinase and stimulated by insulin and allosterically stimulated by citrate. So, as we know, Malonyl-CoA inhibits beta-oxidation of fatty acids, and for example, during exercises, massive amount of ATP is degraded to AMP. So the concentration of AMP increase, it inhibits the activity of this enzyme, thereby it causes decrease in Malonyl-CoA level that results in decrease in fatty acid synthesis, thereby preserving energy because during exercises there is no need to waste energy for fatty acids production. Moreover, organisms have to produce additional energy. So simultaneously, decrease in Malonyl-CoA production induces increase in the activity of carnitine shuttle, thereby it increases the rate of beta-oxidation that results in energy production. So in conditions when tissue requires additional energy, this regulation decreases energy consumption and increases energy production. To explain why citrate stimulates acetyl-CoA carboxylase, we have to know that citrate is the intermediate metabolite in the Krebs cycle, and Krebs cycle has the major regulatory enzyme, isocitrate dehydrogenase, that converts isocitrate to alpha-ketoglutarate, and the activity of isocitrate dehydrogenase is stimulated by AMP and inhibited by ATP, which is logical because the function of Krebs cycle is to prepare metabolites for electron transport chains that will produce energy. And in the well-fed state, when the energy stores in form of ATP molecules are full, tissue does not need additional energy. So high level of ATP inhibits isocitrate dehydrogenase enzyme, and this causes increase in citrate level in mitochondria, and increase in citrate level allosterically stimulates the activity of acetyl-CoA carboxylase that increase production of Malonyl-CoA that inhibits carnitine acyltransferase 1 enzyme, thereby it causes decrease in beta-oxidation of fatty acids. In terms of insulin, right after high carbohydrate meal, when insulin level is high, a lot of glucose molecules enter into the tissue. Then by glycolysis they are degraded to pyruvate, and then pyruvate is converted to acetyl-CoA. So a lot of acetyl-CoA must be stored somewhere, and there is no better storage form than triacylglycerols. So insulin by stimulation of acetyl-CoA carboxylase stimulate production of fatty acids, and then fatty acids bind to glycerol with production of triacylglycerols. And one of the intermediate metabolites in fatty acids production is Malonyl-CoA. So the level of Malonyl-CoA increase, and this inhibits carnitine acyltransferase 1 enzyme, thereby beta-oxidation decrease. So it's the mechanism how insulin through Malonyl-CoA inhibits beta-oxidation. In our organism we have 4 fatty acyl-CoA dehydrogenase enzymes, and each dehydrogenase works on different chain lengths. but they work in a similar fashion. Initially, carbon in the beta position is oxidized by FAD, then molecule is hydrated, and then again carbon in the beta position is oxidized by NAD, then binds coenzyme A with simultaneous release of Acetyl-CoA. Because each time oxidation occurs in beta carbon position, this process called beta-oxidation. So during one cycle, one molecule of Acetyl-CoA released, thereby fatty acid becomes 2 carbons shorter, and one FADH and NADH are generated. What we have to understand is that FAD and NAD function as electron acceptors. Basically, each of them take away 2 electrons from fatty acid and transfer electrons directly to electron transport chain. Recall that on inner mitochondrial membrane located electron transport chain that is composed of 4 protein complexes and coenzyme Q, and also there is 5th protein complex called ATP synthase, and the concept is that protein complexes in electron transport chain transport electrons from one complex to another, and by this electron flow they produce energy to drive hydrogen protons at the level of complexes 1, 3 and 4 from mitochondrial matrix into intramembrane space. This creates hydrogen proton concentration gradient across inner mitochondrial membrane with high hydrogen concentration in intramembrane space and low concentration at the mitochondrial matrix. And ATP synthase uses this concentration gradient, and by transportation of hydrogen ions from intramembrane space into mitochondrial matrix it generates energy that results in production of ATP molecules. The major difference is that FADH transfers electrons to complex 2, and this results in production of 1.5 ATP molecules per 1 FADH molecule, and NADH transfers electrons to complex 1, thereby electrons will undergo one additional step in respiratory chain, and this will create higher hydrogen proton concentration gradient, and because of that it will result in production of 2.5 ATP molecules per 1 NADH molecule. So basically dehydrogenases make substrates for energy production and shorten fatty acids molecule. Obviously, the higher the chain length of the fatty acid, the more cycles of dehydrogenation it undergoes, the more ATP molecules are produced. And the important concept to know is that mitochondria is capable to oxidize only long chain and medium chain fatty acids, no more than 20 carbons. Very long chain fatty acids that have more than 22 carbons have to be shortened, and this shortening is provided by peroxisomal beta-oxidation. The logic here is that because mitochondria do not have enzyme to oxidize very long chain fatty acids, they are initially delivered to peroxisomes where they undergo peroxisomal beta-oxidation. Peroxisome shortens them to medium chain with simultaneous release of Acetyl-CoA and binds to them CoA molecule. So peroxisomal beta-oxidation results in production of medium chain length fatty acyl-CoA. And now medium chain length fatty acyl-CoA is delivered to mitochondria where it can undergo normal beta-oxidation. Very high yield to know that this process in human body occurs strictly in peroxisomes, because if something wrong with peroxisomes, no other organelle in human body is able to oxidize very long chain fatty acids, so they will simply accumulate in tissues. And at the end of beta-oxidation there are 2 possibilities. Most fatty acids in our human body have 16 or 19 carbons, such fatty acids called even chain fatty acids. In this case, if each time 2 carbons are cleaved, it leaves fatty acid with 4 carbons, that is split into 2 Acetyl-CoA molecules, and Acetyl-CoA can be used in Krebs cycle for energy production. But some of the fatty acids have 17 or 19 carbons, such fatty acids called odd chain fatty acids, and in this case beta-oxidation leaves fatty acid with 5 carbons, that is split into Acetyl-CoA that goes into the Krebs cycle and propionyl-CoA. Propionyl-CoA is then carboxylated by propionyl-CoA carboxylase to methylmalonyl-CoA. Important to know that propionyl-CoA carboxylase is one of 3 carboxylases that use biotin as coenzyme, same as pyruvate carboxylase in gluconeogenesis and acetyl-CoA carboxylase in fatty acid synthesis. Methylmalonyl-CoA then is converted by methylmalonyl-CoA mutase into succinyl-CoA. Methylmalonyl-CoA mutase is one of the 2 enzymes in human body that use vitamin B12 as coenzyme. Another one is methionine synthase. And succinyl-CoA then goes into the Krebs cycle with subsequent energy production. We have two groups of disorders that affect this metabolic pathway. The first group are disorders of beta-oxidation, and the second group are disorders of utilization of propionyl-CoA that are called organic acidemias.